very long one. I didn't ask for it. I should have asked for CV. But I got it. Good morning. Good morning. Well, this is the start of our fall semester of seminar, so thanks for coming. I'm Dennis Zakowski. I'm the Science Director here at the Foundation. We have here in the room, I guess, about 25 people or so, and on our webinar we have another 30 or so people. So there's an interest in the subject of today's seminar. Just a little bit of logistics. Anyone in the room that needs to use the facilities, we've got keys up at the desk, and there's restrooms down at the end of the hall beyond the elevators. Those of you that are on the webinar that have never been on before, I think you can see that you have the ability to see and hear us, but you can't talk to us. You can ask questions later on in writing, and Chester will take those at the end of the talk. Today's talk is called Understanding Nitrogen Transformations and Removal at Oyster Restoration Sites in Hudson River Estuary. As a little bit of background, a few years ago, the Foundation, principally through the work of Jim Lodge, got involved in a project involving determining whether oyster restoration in the harbor was feasible. And there was a series of actions that Jim and company laid out that were necessary to try and make that determination. A number of pilot sites were set up around the harbor. We were interested in looking at a couple of key questions. We wanted to know whether indeed bringing oysters back into New York Harbor made sense ecologically. We thought that they're interesting ecosystem engineers and they would provide habitat value. And we also were wondering whether or not, or how much water quality benefits they might produce. So we're interested in whether or not oysters could indeed transform nitrogen, essentially remove nitrogen from the water column and put it in other parts of the ecosystem that might wind up with water quality improvement. So our request for proposals went out a few years ago, and we're happy to select Chester Zarnoch and Jim Holine as principal investigators to examine that question. So the work is completed, and we're pleased to have Chester here to talk about it. Chester is an assistant professor at Baruch College. He's working on a variety of issues of marine ecology, including aquaculture, biogeochemistry, and he's been a great addition to the metropolitan area, and we're glad to have him here. Jessica.
Okay, so I'm going to talk to you guys about the work that we've been doing on oysters and nitrogen cycling throughout the Hudson-Raritan Estuary, the Hudson River, Easter River, Jamaica Bay. And over those years, we've collaborated closely with Tim Holine, who's at Loyola University, Chicago. So him and I have been working on all things nitrogen-related for the last several years. We had the good fortune of collaborating with some great folks. We had a graduate student as part of the Hudson River Foundation grant, Mike Hassick, who's at Loyola, Denise Brusewitz, who's now at Colby, Brett Bronco at Brooklyn College, Allison Fitzgerald, who was a PhD student at CUNY, now assistant professor at New Jersey City University, Wayne Gardner at University of Texas, and Ray Grizzle up at the University of New Hampshire. Hopefully I can really make this sort of interesting as the biogeochemistry of nitrogen could possibly be. And we can make this an enjoyable. As Dennis mentioned, there's about 30 people in the room about 10 minutes ago, but for those of you that are on the webinar, we probably had another 30 or more people have entered the room since. It's a rambunctious crowd. It's a rambunctious crowd. And everybody's really excited to hear about oysters, right? Right. So I probably have to change my presentation. There's no one's here to talk about nitrogen, but But really what we're going to talk about is sort of that relationship of oysters and nitrogen in the Hudson-Raritan Estuary. And even though there's lots and lots of interest on oysters here in New York City, nitrogen is debatably the biggest environmental concern that we have to deal with here in the Hudson River system. Bob Howard published a chapter in the Hudson River Estuary book showing that the Hudson River Estuary is one of the most heavily nitrogen-loaded systems across the globe. Nitrogen loading leading to eutrophication, excessive organic carbon production in estuaries is common. Generally, you see N limitation in estuaries, and that's generally due to a balance of nitrogen fixation and denitrification. But when you have this excessive load of nitrogen, the systems shift to becoming eutrophic. And in New York, the major contribution to this end load is our sewer systems. We have this combined sewer system where all the storm water running off New York City streets, the concrete off New York City streets, goes down our storm drains and gets combined with the wastewater from our homes and businesses, and would normally go to a wastewater treatment plant. However, when we have excessive rains, or rains and heavy rains in short periods of time, the volume of water running off the streets, going down storm drains, coupled with the wastewater from our homes and businesses, is too much for our wastewater treatment plants to handle, and so that wastewater is diverged and released into our estuaries. And there's about almost 500 points where we're releasing this untreated wastewater into the system throughout New York City. And so this figure here shows all the locations of those outfalls. Okay, we have about 14 wastewater treatment plants scattered throughout the 5 boroughs, and 500 or so of these outfalls. And the outfalls are releasing 100 million to 1 billion to 2 billion gallons of raw sewage into the estuary each year. So billions of gallons of untreated water going into our system. And so here's a picture of a CSO on the Brooklyn side of the East River, really large CSO. So when we have this massive nitrogen load, it leads to an increase in primary production. Primary production is going to, the increase in primary production is going to manifest itself into really turbid waters due to phytoplankton, high densities of phytoplankton, and also you have these large macroalgae blooms. And so this is a shot of one of our study sites in Jamaica Bay in 2009, Spring Creek. And you can see the intertidal zone is just littered with this ulva, this is sea lettuce. And this is in July, and a couple of months later, in September, after that organic material starts to decompose, what we see is a massive die-off of soft shell plants, Maya area. You can see lots of the shells here. Take a close-up. You can see the Maya, as well as lots of mud snails, very high density of mud snails, and you can even see a film of sulfur bacteria on the surface of the intertidal sediments. And so this is a rare case, but was very significant. This is the subtidal. Again, you can see all of the shells, and you can see the shells located throughout the entire creek. And so these kinds of die-offs, due to sediment anoxia, fish kills in creeks that are not well flushed, is one of the common symptoms of a heavily eutrophic system, particularly a system like embayments that aren't completely well flushed, like this portion of Jamaica Bay. So it brings us to our oysters. And so as Dennis mentioned, there's a couple of ways in which we can figure that oysters may play a role in influencing water quality. The first, and one that we probably know the best, is that their role in water filtration. Their role as a filterer of the water. So oysters have the potential to filter very high volumes of water. You heard numbers like 50 gallons per day, 7 liters per hour, very high potential filtration rates. And this can have an impact locally on a small scale around an oyster reef, and there's been experimental work to demonstrate that. It can also have an impact at ecosystem level scales if you think about zebra mussels throughout North America, including the Hudson River, Corbicula and the Potomac River, and so on. There's lots of examples where you can have this ecosystem level reduction in phytoplankton due to the filtering capacity of oysters and other bivalves. So clearly water filtration is an important service that oysters may provide in a eutrophic system like the Hudson-Raritan estuary. Now the other piece of it is the other important potential ecosystem service is through enhancing coupled nitrification, denitrification. So generally the microbes in the sediment are fed nutrients through the settlement of organic matter out of the water column. So phytoplankton settling down, algae settling down on the bottom, and then getting mineralized. Okay, and so that organic material, it gets settled and then can potentially go through a series of transformations that can ultimately lead to the removal of nitrogen from the system, converting organic nitrogen in the form of sea lettuce, phytoplankton, into an inorganic form of ammonia through ammonification. Ammonia gets converted into nitrate through nitrification, and that nitrate can get removed from the system through denitrification. Now, with oysters, the idea is that you're going to take that settling organic matter, that phytoplankton that normally is just falling on the bottom, and you're going to package it. Okay, it's going to get filtered by the oysters and sort of packaged into these structures that we call biodeposits, which are their waste. And these biodeposits, they actually have two kinds. They have feces, which is material that goes through their digestive processes, and then you have pseudofeces, material that is rejected by the oyster without putting it through the digestive processes. So these biodeposits are essentially what oysters have filtered out of the water. Those particulates, organic carbon, nitrogen in the form of phytoplankton, and other organic material that might be in the water column, or inorganic material that might be in the water column, and it's centering it down to the sediment. It's funneling it down. So it's sort of acting like a conveyor belt and enhancing that sediment process. And so if the biodeposits can enhance the amount of organic nitrogen, more of that organic nitrogen lead to more ammonification, lead to more nitrification, leading to more denitrification, which is what in a eutrophic system we're interested in seeing, because we're removing reactive forms of nitrogen and converting it into non-reactive, inert nitrogen gas. We okay with that? So it seems like a pretty linear process, but it's anything but. It's really complex. It's really nuanced. There's a lot of environmental factors that come into play that control things like mineralization, ammonification, nitrification, and denitrification. So, for example, nitrification is performed by a certain suite of bacteria, chemolithotrophs, that use inorganic carbon and oxygen to convert ammonia into nitrate. So they're aerobic, and they're using the inorganic carbon as a source to do this, to perform this process. While the denitrifying bacteria are anaerobic bacteria. So they generally don't live where there's oxygen, and they require an organic source of nitrogen in order to perform this conversion of nitrate to nitrogen gas. So the suites of microbial organisms that are performing these tasks have different environmental requirements. And that plays a very important role in trying to dictate whether oysters are mediating this process. If oysters are really pushing the organic nitrogen in the water column into an inert form of nitrogen. So about the time that we were working in Jamaica Bay, 2010, 2011, some other folks were working on this down south. Mike Peeler, Ashley Smith published a really nice paper in 2011 where they measured denitrification rates in various habitat types in a North Carolina system. And what they found is that oyster reefs, sediments adjacent to oyster reefs, had among the highest denitrification rates when compared to any other habitats. And they argued that this high denitrification rate in the summer was likely due to benthic-pelagic coupling. They're feeding more, they're depositing more organic carbon and nitrogen to the sediment, and then that's driving coupled nitrification, denitrification. And then later on, a couple other papers came out. These are papers that came out in 2013. And one paper, Higgins et al. Found what these folks did is they measured denitrification rates under aquaculture cages. And so these are floating cages in parts of the Chesapeake. And they were measuring denitrification rates below the cages. The idea being those biodeposits are going to land on the sediment and then fuel the microbial processes. And they found no effect of oysters, no effect of oyster biodeposition on denitrification. While another paper, also down in the Chesapeake, Kellogg et al., found very high rates of denitrification using a method where they essentially incubated an intact piece of reef and measured denitrification rates on a reef structure itself. And the rates that they found were among some of the highest rates, these are millimoles of nitrogen flux, some of the highest rates of denitrification ever observed.
That's a lab study?
This was a field study. So they took a piece of a reef, they put it into sort of like a cylinder, put it back on the reef, let it settle for a while, and they came back, sampled it, brought it into the lab into a mesocosm, a microcosm, and then conducted their measurements.
The Higgins study, were oysters in the cages, or were oysters living down below the cage?
The oysters were in cages. So they were floating cages, a typical method used to grow oysters down there, and they were measuring it below. So the effects of oysters on denitrification are somewhat mixed and somewhat nuanced. There's some contextual basis that needs to be understood in order to sort of decipher the true role of oysters on denitrification. So that brings us to New York, in oysters in New York. And so we know that New York has a long history of oysters. Oyster was, at one time in New York, oysters were sort of the kingpin here in New York, the oyster capital of the world. 100 million oysters eaten in one season. This is 1904. Oyster barges were located throughout the city. They were as common as hot dog stands. As we see now. But by 1920s, overfishing, habitat loss due to dredging and hydrological modifications led to the demise of oysters, contributed to the demise of oysters, and pollution was probably the key factor that led to the loss of oysters, eutrophication, and associated with sewage releases, and that's some work that Dave Franz did back in the 80s. But over the last couple of decades, we've seen significant improvements in water quality throughout the Hudson-Raritan Estuary. And there's been interest in whether or not we can bring oysters back. There's been a number of studies over the last couple of years that show the potential for oysters to grow, survive, reproduce successfully throughout the estuary. And oysters have then sort of been integrated into management plans, creating a certain number of acres of oysters over the next several years to decades. And then the effort led by the Hudson River Foundation, the Oyster Restoration Research Project, to determine the feasibility of oyster restoration by building pilot oyster reefs, is sort of where we're at now. And you know, a big part of I think the interest in oysters is sort of the cultural connection of New York City and oysters. And I think that was fomented by the book The Big Oyster, Mark Polanski. And then there's been documentary films talking about oysters in New York, and now we also have the New York Harbor School and their ambitious plan to put 1 billion oysters into New York Harbor. And since Sandy's, we've also started to see oysters being integrated into plans for ecological resilience. And in the Rebuild by Design competition, one of the winning programs was this living breakwater idea, where rock structures were going to be seeded with oysters, and so oysters were going to be integrated into ecological resiliency plans. So there's lots of momentum, there's lots of interest in oysters in New York City, and so we think that it's worthwhile spending some time looking at the interactions of oysters and water quality. And so we conducted a number of studies over the years to talk about each of these. Initially, our work was focused on looking at how oysters can influence nitrogen cycling in urban systems, and really we wanted to sort of key in on how nitrogen loading influences this overarching question. Okay, and then, if we were going to do oyster restoration, do we need a certain oyster density in order to achieve impacts on sediment nitrogen cycling? And the idea is that these questions could inform managers, folks that are interested in putting oysters out there, so that we can identify locations and optimal densities to have some kind of nitrogen mitigation. So our initial effort was focused in Jamaica Bay. Jamaica Bay is located at the southwest portion of Long Island. Has 4 major wastewater treatment plants, many combined sewer overflows, a combined load of about 16,000 kilograms of total nitrogen per day. That's about 34 or so thousand pounds. And to put that number into perspective, it works out to about, I think, 1,200 pounds per acre per year. And if you were going to fertilize a cornfield, you would probably fertilize at about 150 pounds per acre per year. So a tremendous nitrogen load. Most of that load is coming from the wastewater treatment plants and combined sewer overflows. And the mean depth is about 5 meters. We chose 4 study sites within the bay, and the idea was to create these study sites along the nutrient gradient. So the bay gets flushed from the Atlantic Ocean through Rockaway Inlet, and so you have the greatest flushing near the inlet, and then as you go further back, you have longer residence times, less flushing. So our study sites were Floyd Bennett Field, which was sort of our reference site, closest to the inlet. And then we had our intermediate sites at the Wildlife Refuge in the center of the bay, and then Mott's Base at the east end of the bay. And then our poor water quality site at Spring Creek. So that was the site that I showed you earlier with the ova and the massive die-off of the Maya-Aran area. And so just to remind you, Jamaica Bay is a tremendously productive ecosystem. And so this is just a plot of chlorophyll A over the 2-year study period. We worked in Jamaica Bay, and you can see that there are blooms of phytoplankton well over 100 micrograms per liter throughout our sampling period. And in most cases, it's ranging 20 to 50 micrograms per liter, so well above the EPA criterion for poor water quality. And so this is the conceptual diagram that's sort of going to guide the talk. The idea is that, again, as I mentioned, we wanted to look at the influence of oysters on nutrient cycling, nitrogen cycling across a gradient of N loading. We're doing that by having these 4 sites across that gradient. And then at each site, what we're going to do is deploy oysters at different densities in cages over sediment trays. So similar to what the Higgins et al. Study did. And the idea is that those oysters in those cages are going to release biodeposits that's going to influence the sediments below, the sediments in the trays below. we don't have, in Jamaica Bay there are no naturally occurring oysters, so we couldn't take advantage of a natural reef for oysters that are already existing there. So we had to use this sort of experimental design of boxing cages. And again, what we're looking for is for the oysters, and so this is our oysters here, oysters to increase organic matter, leading to the ammonification of that organic matter to ammonia, to nitrate, to inert nitrogen gas. And so we measured, the sediment organic matter, the amount of ammonia in the sediment, the ammonification rate conversion here, nitrification rate conversion nitrate to ammonia to nitrate, and then denitrification. We measured denitrification potential, so a way of estimating denitrification. It's not a direct measure of denitrification. It's called the acetylene inhibition technique. And so what that allowed us to do is increase our replication, both of samples, but also spatially and temporally, using that technique as compared to other ways of measuring denitrification more directly. And so this is what our boxing cage design was. These are sediments. Sediments were covered with a tray, with a top, with a cage, and then the oysters were stocked in these cages at different densities. A low density of 40 per square meter, intermediate density of about 80 per square meter, and a high density of about 150 per square meter.
What's the distance between the oysters and the tray?
It's about 6 centimeters. And so the trays were deployed subtidally at each site along a trot line. And then we would sample these trays bi-monthly, take apart the cages, collect our sediment samples, bring our sediment samples back to the lab where we can measure each one of these processes. So I'm going to start by talking a little bit about our measurements of these processes in the sediment, and as we go through it, sort of walk you through this pathway. So the first thing that we saw was that there's a date effect. So as we measured the amount of organic matter in these boxes below the cages, it changed through time, and that wasn't necessarily surprising. But there was also a treatment effect, an oyster treatment effect. And so if you look at these plots, this is sediment estuary dry mass. The open circles are our high-density treatments. The dark closed circles are our controls, no oysters at all. And you can see for most of our sites, the highest have the greatest estuary dry mass, while the controls have the lowest. We had one exception at Mott's Basin where that didn't exactly hold true. But over the course of 2 years, we can say that the oysters are enhancing organic nitrogen in the sediment. So we're seeing that first link in this process. And so the next question is that organic nitrogen getting converted into ammonia? And so what we looked at is the relationship of organic matter to exchangeable ammonia, a way of estimating pore water ammonia. And we see this relationship, a significant relationship across sites and seasons. And so, the next link looks good. Okay, organic nitrogen going into ammonia.
The biodeposits are both pseudo and regular feces.
Right. Combined. Yep, the biodeposits are exact, feces and pseudofeces. And then we want to look at ammonia getting converted into nitrate through nitrification. And then this is where we run into a problem. So what we're looking at here is nitrification rate in relation to exchangeable ammonia, the amount of ammonia that's in the sediments. And we have a very weak relationship, not a lot of the variation explained. So nitrification and pore water ammonia not related. And then water column nitrate and nitrification not related. And so what's happening here? If we look at the relationship of ammonification and nitrification, we can start to understand what's happening, or why there's sort of a roadblock at the nitrification point. When we look at nitrification in relation to ammonification, we see that a large number of our samples are showing what we call immobilization. That ammonia is getting recycled, getting uptake and assimilated by heterotrophic bacteria. It's not getting converted into nitrate by nitrifying bacteria. And that's common because nitrifying bacteria aren't the greatest competitors for ammonia. So if we were to just remove where we have this immobilization, where that heterotrophic uptake of ammonia is dominating, and we only look at the positive side, nitrification, net ammonification, we do see this relationship of ammonification and nitrification, where the ammonia in the water column, the process of producing ammonia in the water column is related to the process of nitrification. Now this kind of heterotrophic uptake of ammonia is very common in eutrophic systems, highly organic sediments. Okay, and lastly, the last part of that link is this denitrification, and with denitrification, we see that there's time differences across our study period, but there's no treatment effects. So our oyster treatments, low, high, and intermediate oyster densities, have no impact on denitrification. So we're sort of able to understand what's happening here. We see nitrification is limited. Why aren't we getting denitrification? And one reason is because nitrification and denitrification are not related, and that would be expected if immobilization is being favored over nitrification. Okay? That ammonia that is being recycled by the heterotrophs rather than getting converted into nitrate, which is usable for the denitrification. Remember, the denitrifying community requires organic carbon and nitrate. Right? That's sort of the environmental conditions that allow us to happen. So if we're not getting this relationship of nitrification, denitrification, what's driving denitrification? And what we see is that denitrification is highly related to the organic matter in the sediment, organic carbon in the sediment. Okay, denitrifiers require that organic carbon. And it's also related to the amount of nitrate in the water column. Okay, so the denitrification activity is related to water column nitrate. So what's happening here is that the denitrifying community is not relying on nitrification to supply its nitrate supply. Instead, it's pulling nitrate that's diffusing out of the water column, that's coming out of the water column, and using that as a nitrate supply. And it has access to plenty of organic carbon from oyster biodeposits. We did a nutrient limitation study, so manipulating the amount of nitrogen and carbon in these denitrification assays. And one of the things we found is that the response of denitrification was most significant with an oyster effect when nutrients were limited. Okay? What we found generally is that the sediments were not nitrate limited. Jamaica Bay, plenty of nitrate. I'm sorry. The sediments were generally nitrate limited. They were always pulling in the nitrate because of their requirements to support that nitrate because it wasn't coming from nitrification. And secondarily limited by carbon because Jamaica Bay and the biodeposits are providing so much carbon. So an important point here is that the potential for a denitrification effect in the eutrophic environment is going to occur where you have limitations in nitrogen and carbon in the sediments. Okay? Limitations in nitrogen and carbon in sediments can lead to the potential for an oyster effect. So, to sum up what we found in Jamaica Bay, in order to make this connection of oyster biodeposits, oyster waste to denitrification, we need to follow this process. Okay, the organic nitrogen from biodeposits gets ammonified to ammonia, to nitrate, to nitrogen gas. But that's not what we're seeing. See, the oysters increase the organic matter, they increase the amount of organic carbon and nitrogen in the sediments. And then when you have ammonification, that ammonia generally gets immobilized, and so you don't get nitrification. With the organic carbon in the sediment and the amount of water column nitrate that's available in Jamaica Bay, the denitrifying community is using those two sources to drive denitrification. Okay? So the water column nitrate rather than nitrification derived from nitrate derived from nitrification is most important. Okay. So we in Jamaica Bay in our study, our sort of experimental box and cage design study, we didn't see this oyster effect. But gosh, there's these other results that are out there that show there is an oyster effect, and these guys measured nitrogen denitrification at sediments adjacent to reefs in North Carolina. These guys are measuring denitrification of reefs, literal pieces of reefs in the Chesapeake. So maybe it has something to do with reef structure. Okay, some aspect of reef structure that's really important in driving denitrification. And so that was sort of a burning question for us, and we were fortunate that things worked out, that we were able to collaborate with the Hudson River Foundation and their work to put these pilot oyster reefs throughout the Hudson River Estuary in order to look at the potential for reef success and ecosystem services associated with those reefs. And so it led to our study where we were going to focus in on studying nitrogen cycling at a couple of the oyster reefs that the Hudson River Foundation created, the ORP group created, I should say. And what we were going to do is we're going to use a bit of a different technique. We were going to measure sediment adjacent to the reef. The idea is that those sediments are most likely going to receive oyster biodeposits and sort of charge those microbial processes. And then we're going to compare that to sediments further away from the reef, those that were unlikely to be influenced by sediment biodeposits. And we were going to take into consideration things like oyster biodeposition, the hydrology of the site, measuring our nitrogen dynamics. And we were able to add a couple of different measures that we weren't able to do in the Jamaica Bay study through our collaborations. And one of the key things that we changed is that we changed the way we measure denitrification. So now we're using a method, it's called membrane inlet mass spectrometry, that allows us to directly measure denitrification rather than estimate using the enzyme assay. And another unique piece that we added is this method that is termed the CAN method. And what we do is we take these cores of sediments near the reef, away from the reef, or we even did some studies where we took parts of the reef, and we basically create this microcosm where we flow site water through the cores. So the advantage of this is that we're able to more closely represent what's happening in situ, okay, by flowing water. Generally, when you do these kinds of measurements, you have these closed cores, they're static, and as nutrients change, cycles within those cores sort of change. By having these flow-throughs, I think we're better able to capture in situ type conditions. What it also allows us to do is that we can add isotopes to these things and use the isotopes to trace the nitrogen. So rather than just measuring every single transformation, we can label and measure the transformations of nitrogen through the sediments. And again, all along, really, we wanted to be able to quantify denitrification. We wanted to quantify denitrification, oyster effect on denitrification in Jamaica Bay. But a real key that we sort of focus on in our work is understanding the mechanisms. Okay? Why? Not just how much, but why? What's causing it? What are the mechanisms driving these rates? And by using this CAN method, we really are able to explore some of these things. So most of our work is part of the ORRP study was focused at the Soundview Reef. So here's a shot of the reef. The reef was stocked a few times over the course of our study. In particular, right before our first set of measurements, which happened in July 2011, where there was essentially a density of oysters of around 3,000 per square meter. Excuse me. And these are spat on shell, so these are relatively small oysters. So we don't have a whole lot of biomass on this reef at the time. And so we took sediments, samples, core samples around the sediments, as well as a few reef samples. We had some reef samples that actually had spat on shell inside the cores, and then some reef samples that had no spat on shell. It was just shell in the cores. And we brought them back to the laboratory and set up these incubations. And again, we're running site water through these cores in order to perform our measurements. And so here's a sediment core, and here's a shell core. And so what we found. So what we're looking at here is denitrification through time. Is our summer sample, a fall sample, spring sample. Near the reef, adjacent to the reef, where we would expect to see some oyster biodeposition, and far away where there wouldn't be. And then here what we're describing is a potential denitrification. So this is when we add our 15 nitrate tracer. So we're sort of increasing the amount of nitrate that's in the water column. And what we see is that there's no effect of the reef in any of our seasons. Summer, there's a tendency to have greater denitrification, potential denitrification, tendency. But this really isn't all that surprising given the biomass on the reef. It's a relatively young reef, just stocked. Not a lot of biomass on there, not a lot of potential for high oyster biodeposition, but we do see sort of a trend. When we use our tracers in order to distinguish between coupled nitrification, denitrification, okay, denitrification being fueled by nitrification, and then water column fueled denitrification, which we term direct denitrification, we see that by far most of our denitrification is being driven by that water column nitrate. Okay, not a lot of coupled nitrification, denitrification. So that supports what we found in Jamaica Bay. And then when we look at our spat on shell versus just our shell with no spat at all, again we see a trend towards higher denitrification in the spat on shell, both for denitrification and potential denitrification, but it's not statistically significant. Here's something that's interesting, though. If we look at the amount of denitrification in our control cores as compared to those that were amended with 15 nitrate, the amount of denitrification is similar. We go back to our sediment cores, the amount of denitrification around 400 or so micromoles per square meter per hour. Our potential denitrification about the same. So what this is telling us is that the denitrifying community is not nitrogen limited. Okay, it's not increasing denitrification with more access to nitrogen. So carbon may be the key here. And when you think about a reef becoming more developed, okay, having more life, more things living on it, more oyster filtration, biodeposition, carbon deposition, carbon accumulation within the reef structure, around the reef structure, is likely to increase. Whether or not that additional carbon is going to drive this to being a significant relationship remains to be seen. There's potential for it. So we spent a lot of time talking about this side here, trying to understand oysters' role on nitrogen cycling in the Hudson River estuary, Hudson-Raritan estuary. I'm going to spend a few more minutes talking about water quality parameters. Okay, oyster filtration. And so our approach to measuring oyster filtration is something called the biodeposition method. And the premise of the method is that you characterize all the particles that are in the water column, the system, okay, the organic and inorganic particles, and then you measure the biodeposits. So we actually stand there and for hours and hours on end collect oyster poop, and as exciting as boring as that part is, it's really exciting to take that information and train it into all these really neat calculations in oyster physiology and feeding behavior. And so, a couple of take-homes of what we see on our oyster feeding data in Jamaica Bay, one of the strongest predictors of clearance rates is the concentration of particles in the water column. Okay, as the concentration of particles in the water column increases, there's a decrease in clearance rates. And so our rates range from about 0.4 liters per hour per gram dry weight to around 4. And we've heard of rates upwards of 7 liters per hour. So lower than most predicted. And you can see that by far a lot of our measurements are at higher concentrations of total particulate matter. This isn't a novel result. There's been lots of studies done on oysters that show this negative relationship with total particulate matter and clearance rates. And generally it's around 25 milligrams per liter where you start to see the decline. We see a decline a little bit earlier. But I think what's interesting is that in most places, as total particulate matter increases, it's due to more sediment influx. But that's not the case in Jamaica Bay. In Jamaica Bay, most of the particles that are in that total particulate matter are organic material. So we're looking at organic content of the TPM versus total TPM. Most of our Jamaica Bay measurements are around 40% organic material, while other studies is much less. And again, that's because in Jamaica Bay we have tremendously high primary productivity, very high phytoplankton abundance. And so most of what those oysters are encountering are phytoplankton cells. And whether it's a phytoplankton cell or a sediment cell, the oyster responds to it when there's too many of them by reducing its clearance rate and increasing rejection rates, pseudofeces. At Soundview, we were able to do some oyster feeding measurements by putting adult oysters out at the reef. We deployed them in these cages. We put them in the shells of these cages in order to simulate the height of the reef. If we look at total particulate matter through time at Soundview, generally total particulate matter is high at Soundview, okay? Just like Jamaica Bay. But unlike Jamaica Bay, most of that particulate matter is inorganic material. Okay? If you look at phytoplankton abundance at Soundview, most of our measurements are below 10 micrograms per liter. And so this is sort of a paradox. Because you think of a place that's very nutrient loaded, highly nutrient loaded, you would expect to see great primary productivity, but that's not the case. There's been some work at Stony Brook that suggests it's possibly due to the advection of the phytoplankton. There's been some work at a Noah Milford lab that suggests it's light limitation, whatever it may be, but there's just not a whole lot of production in this area. And we see a very sharp decline in organic content with the TPM, with most of our measurements around 20% organic content compared to Jamaica Bay of 40% organic content. And so we took measurements 5 times at Soundview, and for the most part our measurements were less than around 2 liters per hour per gram, similar to what we found in Jamaica Bay, with one exception. In July 2011, we had this tremendous feeding rate, over 7 liters per hour at Soundview. And so that's about as high as they're going to go. And when we try to figure out why we have this high clearance rate, well, it just so happens that this time, July 2011, we had very low total particulate matter, less than 5 milligrams per liter, very high organic matter, and that high organic matter was largely this nice phytoplankton bloom that happened here. Now this, over a 2-year period, never happened again. So it was sort of an oddity. But oysters' feeding behavior responded quickly and took advantage of that bloom in these conditions. So one thing to take out of this is that things are really variable when we think about oyster filtration rates throughout the Hudson River Estuary. And to demonstrate that, we're going to show you some data that we have from basically about a 10-day window in July 2011, where we measured feeding rates at 6 sites throughout the estuary. 4 in Jamaica Bay, 1 in the Hudson River, Hastings on the Hudson, and Soundview. And the range was from 0.9 liters per hour up to almost 7.5 liters per hour. And part of that is explained by total particulate matter in the water column. But a take home here is that if we went out and measured feeding rates once at all these sites, and we found this great measurement at Soundview, we'd say, well, okay, this is the place that we're going to put all our oysters and we're going to clean up the estuary. And if we just did it this once at Jamaica Bay Wildlife Refuge, we say, this is the worst place that we'll ever. And we need to have this sort of temporal and spatial replication because of the dynamics assessed. So benthic pelagic coupling, so water filtration of oysters is possible under the right testing conditions. How are we doing with that?
Oh, good.
Good? Okay. So the next step for us was to sort of to put these pieces together, sort of start to link our feeding measurements with our nitrogen cycling measurements. And think about also how things might be different if we were to look at oligotrophic systems as compared to eutrophic systems like the Hudson-Raritan. And so in order to do that, we had to move outside the Hudson-Raritan estuary. And we ended up collaborating with Ray Grizzle to do a study in the Great Bay estuary where we measured oyster filtration, biodeposition, nitrogen cycling at 2 sites in the Great Bay estuary. One, the mouth of the Squamscott River, which is influenced by a wastewater treatment plant, so it's more eutrophic. And then another spot, which is called Nanning Island, which is very well flushed, generally oligotrophic. And our approach was essentially the same as what we did in Jamaica Bay and the Bronx River. We performed our feeding measurements, our biodeposition measurements, and then also using our core incubations with our ammonia and nitrate isotopes in order to follow the transformations in nitrogen. And so what we found is that denitrification rates were higher at the eutrophic site. So here we're looking at denitrification, Nanny Island being the oligotrophic, Squamscott Island being the eutrophic, and adjacent to the reef, cores collected adjacent to the reef versus those that are away from the reef, which would unlikely be influenced by oyster biodeposits. And so we see that there's this significant interaction of site by reef, and that's because the Squamscott Reef, we're seeing much higher rates of denitrification adjacent to the reef as compared to away from the reef. And so here now we have a result that sort of matches up with the Peeler and Smith and the Kellogg et al. We're getting this higher denitrification rate associated with the reef. And respiration rate was higher at the Squamscott River, and that would be expected because you have more organic matter due to the higher eutrophic conditions. And so here we're looking at the results of the isotopes, and what we see, similar to what we saw in Jamaica Bay, similar to what we saw at Soundview Park, direct denitrification, water column derived denitrifications favored over coupled denitrification. So here, in the 15 ammonia treatment, we see very low coupled nitrification, denitrification at all sites. And then in our plus nitrate treatment, we see much higher rates of of denitrification adjacent to the reefs, both sides. So there is this reef effect with the water column nitrate, both sides. So again, the sediments here are responding to that plus nitrate treatment. So the questions we had is, okay, well, why? Again, going back to mechanisms, can we explain why we see these differences? And, one of the reasons we came up with is that there's some differences in the feeding and biodeposition that's occurring at the two sites. If we look at chlorophyll at these two sites, we did what we call size fractionated chlorophyll A. So we measured total chlorophyll, the amount of chlorophyll that's in a 5 to 28 micron fraction, and that's like an ideal size for oyster capture. They can really capture that size range well. And then a less than 5 micron, which would be a size range where the oysters are not very efficient at capturing those cells. And chlorophyll was higher at the eutrophic site as expected. But what's interesting is that at the eutrophic site, most of the chlorophyll, most of that phytoplankton assemblage was of a good size for oyster filtration. While at the allotrophic site, most of it is less than 5 microns. So not a good size. And then if we look at things like biodeposition, so total biodeposition, higher at Squamscott as compared to Nanny, the eutrophic site, more biodeposition. The amount of carbon and nitrogen biodeposition, higher at Squamscott as compared to Nanny Island. And then the CN ratio of those biodeposits. CN, being an indicator of the quality of the biodeposits, how labile that organic material is, higher, lower rates at Squamscott, so it's more labile, higher CN ratios at Nanny Island. So the amount and quality of the biodeposits going to the sediments around the reef is greater at the Squamscott River reef, the eutrophic reef, as compared to the allotrophic reef. So oyster biodeposition may be playing into that denitrification effect. The other thing that we noticed is that if we look at the ash-free dry mass of, so we measured the organic matter in the sediments, and we noticed that the sediments, Tim noticed that the sediments were really coarse. There's some coarse material, there's some shell hash mixed in there, and so we fractionated the sediment before doing ash-free dry mass. And what he found was that there was greater organic material associated with larger particles. More organic material accumulated with bigger particles like pieces of shell. And so there was more of that organic material at Squamscott site. And then if we relate that amount of organic material to the amount of denitrification or potential denitrification, we see a correlation, marginally significant correlation, explaining potential denitrification. So it appears that the organic substrate and the potential for enhanced nitrate diffusion. Think about these larger shell pieces, this hash, sort of creating both a biofilm for microbial communities, but also creating a pathway for more nitrate to diffuse into the sediment. They're sort of irrigating the sediments in a way, allowing more nitrate to get in there. So, last thing that we did is say, well, it appears that there's this organic carbon component to everything that we see. Oysters providing this organic carbon, and that organic carbon subsidy driving rates. If we were to compare an allotrophic system into a eutrophic system where you have elevated nutrients in the eutrophic system, elevated organic material in the sediments in the eutrophic system, to a allotrophic system with low organic material in the sediments, low water column nutrients, will we see an oyster effect? Where would the oyster effect be? And so Mike Hassick, the graduate student who worked on the Hudson River Foundation grant, which is Mike right there, conducted this lab experiment where it was essentially a 2x2 design that was replicated, eutrophic, allotrophic with oysters and without oysters. And what it found is that under low trophic conditions, low organic matter in the sediment, lower water column nutrients, the oysters had a significant effect. Okay, oysters increasing denitrification. Okay, so lots of information, lots of data. What does it all mean? We bring it back to what's going on here around New York City now. We have the phase 1 of the ORP completed, moving on to phase 2 and 3 and 4, right, Jim?
6.
And 6. We have they're building an oyster project, and we have potential for building this living breakwater. What have we learned from our work over the last couple of years that can inform some of these future efforts? So first things first, the water filtration. Oysters are most effective at filtering at less turbid sites. This is nothing new. It's a great quote. Victor Zarnoch, pioneering oyster biologist, said oysters feed most efficiently in clean water. Yeah. And you might sort of think of them as sort of as a positive feedback mechanism, sort of keep clean water clean, not necessarily as a way to remediate water. But if there are no oysters out there and we add oysters, we're still removing some nutrients, assimilating some nutrients. That's phytoplankton that could settle in areas that are not well flushed and can become hypoxic. And so we could transfer the movement of organic carbon and nitrogen to areas that are best suited for dealing with that organic carbon through oyster restoration. But one of the things we learned is that you can't go out there and measure filtration rates once at a site, okay? Because it's highly dynamic. Okay, it's going to change based on testing conditions, and so you need to measure it over and over in order to really understand seasonal dynamics as well as the dynamics associated with the system. 2. The mechanism for oyster-mediated denitrification will likely be through carbon biodeposition. And this is really particular for HRE, where we have such high water column nitrogen, high DIN. So with DIN being so abundant, there's going to be sites that the sediments are carbon limited. Okay, areas with better flushing, for example. Okay, coarser sediments. Or, you look at the rebuild by design location, an area that's really good currents. The plan is to promote currents. There's potential for oysters to provide that carbon to drive denitrification rates of water column nitrate. 3, sediments around the reefs or around breakwater structures may have enhanced nitrate diffusion. In our work up in New Hampshire, we found that shell created biofilm, created a pathway for nitrate to diffuse. Some of the things that we've seen with the ORP is that spat-on shell gets pushed off the reefs. And yeah, that wasn't part of the plan, but that may not be necessarily a bad thing because it's adding biofilms, it's adding structure, it's adding a potential for nitrate diffusion. It could possibly be a mechanism to enhance denitrification. And then 4, I think this is the direction that we need to move forward, is that we need to take intact living structures, like the Kellogg work down in the Chesapeake, where we have mature reefs. The ORP is at a point where they're getting, have mature, healthy reefs. We need to take some of those structures and incubate them in these flow-through microcosms using isotopes in order to not only describe the nitrogen cycling, but to be able to explain the mechanisms behind it. And so that's my take-home messages and my advice. And these are our acknowledgments. The Hudson Foundation, of course, for funding our work, National Science Foundation. We worked with a number of different partners, and we were fortunate to have some really great undergraduates and research assistants participate in our projects. Thank you. Questions? Yeah, so that's a good question. And after hours and hours of staring at them, you know. But it's actually really easy to tell in oysters because one side is where the pseudofeces comes out, and the other side is where the feces comes out. Unlike mussels, for example, where they're both shot out the same area.
In the Kellogg study, what they're somehow using some mechanisms to isolate that particular piece of the reef, and then keeping the rest of the reef sort of intact. And then they're measuring things only happen within this microcosm. And then, so that's question one. And then the follow-up would be the carbon aspect of that. It's not just the oysters, but everything else in theory that's living on that reef. And have they measured, the other organisms and try to sort of, take apart that piece from the oyster piece and see which one is really controlling, or is it a combination?
Yeah, good question. So the first part, it's my understanding, is what they did is they took a piece of a restored reef, and they put it in, this cylindrical piece of PVC with a mesh bottom, and then they put it back on the reef and let it sort of acclimate to that period of manipulation, and then came back and took that out, and then brought it into the lab and created this mesocosm where they were able to measure the rates. And what they did is that they actually took apart the reef and identified all the different living components, not just the oysters. Oysters were by far, I think, the greatest biomass, if I remember correctly. But it's clear that oysters are not the only players in this community. It may not just be oysters with it, like, for example, the Rebuild Life by Design, may not just be oysters that are going to contribute to it, but whatever other filtering organisms that are out there that are, creating benthic pelagic coupling that can give organic carbon and nitrogen. So,
yeah. Well, and all the other organisms, like shrimps and
yeah, because they're feeding on that organic material and eating and pooping just like everybody else.
Right.
Nitrification is what you're shooting for in a polluted environment, but to what extent will immobilization benefit, or is it just a temporal?
No, I think immobilization is sort of a negative because the immobilization is just recycling that ammonia. So you don't want to see that ammonia to continue to be sort of cycling in a reactive form.
Maybe we could take the second from the bottom, Lisa Holt's question.
Yeah, so great question. So,
Can you read it over?
sure. Have you considered correlations with benthic and fauna like clams as mechanisms of carbon nitrogen cycling in sediments? So in our Jamaica Bay study, we did a treatment. I didn't talk about it today. We did a treatment of hard clams rather than oysters, and so we had hard clams in the sediments. I'm pretty sure we didn't see a hard clam effect on denitrification, similar to what we saw with the oysters. We also measured other living things within those trays, and we kind of created this artificial environment by having a screen over the top. And so what we ended up finding is that lots of things settled into the trays, probably more than you would find outside of the trays because of the protection of the screen. And so at some sites, like Floyd Bennett Field, our high water quality site, we have tremendously high densities of Maya arenaria, angel wings, and hard clams, other types of bivalves and fauna. And we do see significant relationships with the number of these bivalves and fauna with things like nitrification. Now remember, nitrification is a process that's aerobic, it requires oxygen. So these benthic and fauna are sort of irrigating the sediments, providing oxygen for nitrification.
Looked at what mussels do in terms of the processes?
Sure. So the question is, has anybody looked at mussels? Within the bays of the Hudson-Raritan Estuary, you tend to see rib mussels being the most dominant mussel. In the more flushed areas, the marsh mussels. You see the blue mussels. There's been some work done in the last couple of years on rib mussels right near Soundview Park by the folks at the Milford Lab. And they measured rib mussel feeding rates, clearance rates using the same method that we use, biodeposition method, and showed clearance rates of around 2 liters, if I remember correctly, around 2 liters per hour per gram dry weight. I think part of the attraction of using oysters is there's some cultural connection to oysters. 2 is that oysters can live subtidally, and so they have the potential to filter longer during the day as compared to rib mussels. If you would put rib mussels subtidally, you'd most likely have to provide protection, because without protection, they're susceptible to predation. But there's a lot of interest in rib mussel. Sure. So the problem with blue mussels in a lot of the places that we work is that the water temperature gets too high and they die off. But if you get into the main channels where you have higher flushing or closer to the inlets, you do get plenty of blue mussels. Lots of data on clearance rates on blue mussels, but nobody's looked at it here in the harbor, as far as I know.
Yes, sir. What percentage of the pseudofeces is flushed out from these reefs into the water column?
Yeah, that's a good question.
Can you just repeat it?
Yep, I'm sorry. Thanks for reminding me. So the question is what percentage of pseudofeces gets flushed out of the reef environment, away from the reef, and possibly not into adjacent sediments. And there's been some modeling work done, I think, down from the folks in the Chesapeake region to explore that. We've collected some hydrologic data with Brett Ronco, and so we're hoping that we'll be able to turn some of our biodeposition rates to make and use them to make predictions of what the potential deposition would be around the Soundview Reef. But yeah, it's going to be very site specific based on the currents and the three-dimensional structure of the reef. I mean, that's one of the key things. I mean, when you have that really complex three-dimensional structure, you're creating lots of microsites where the turbulence is reduced and allows deposition.
There's one on the last one there.
In the initial study, in the initial Jamaica Bay study, you suggest nitrifiers are competing for ammonia with other processes, generally mobilization. Given the high concentrations of ammonia, could it be possible that nitrification is low for other reasons? Did you measure oxygen profiles or poor water sulfate concentrations? We did not measure oxygen profiles or poor water sulfide concentrations, but Ames is right that absolutely could be a contributing factor to the reduction in nitrification. Those nitrifiers require aerobic conditions, and in those boxes underneath the sediments, in those sediments underneath the oysters, very, very high potential for anaerobic conditions to inhibit nitrification.
From a public policy standpoint, dealing with nitrogen in Jamaica Bay, and I guess weighing enhancing oyster reefs versus trying to eliminate nitrogen at the plant through other methods. I mean, where does this leave us? It doesn't seem particularly effective here.
Yeah, no. The bottom line is we need to reduce
The question is, what's the...
The question. I'm going to summarize the question and say.
It doesn't matter.
Yeah, does this matter at all in a place like the Hudson River system or Jamaica Bay, where we have so much nitrogen coming from wastewater treatment plants? And the bottom line, when we study all these mechanisms and drivers for all these rates, it's how much nitrogen is in the water. That's driving not just oyster-related ecosystem services, but all other really important ecosystem processes that define the health of the ecosystem in and around the HRE. So the bottom line is we need to find ways to reduce the nitrogen inputs, without a doubt. When we get to a point where we start to reduce the nitrogen inputs, we may be able to see some of these ecological benefits.
Well, I mean, obviously, the other point is that it's not all about removing nitrogen. It's about having
I'm sorry, I didn't hear what you just said.
So it's more than just, the purpose of restoring oyster is is more than just trying to improve water quality and remove nitrogen, but also to bring back that lost habitat value and the associated species.
We just comment with, so however, you made the point, I think, Chester, that the Rebuild by Design project in Lower Bay possibly could have some more benefits, and a lot of the folks in New Jersey that are working in areas like Sandy Hook Bay, where you've got more of the ocean influence, presumably more pollution, less nutrients. What's your take then on those areas?
Yeah, so I think the analogy that I would point to is the Peeler and Smith work, where they worked in Boat Sound, which is highly allotrophic, very low water column nutrients, and all their denitrification rates were related to sediment oxygen demand, which is an indicator of coupled nitrification, denitrification. So in those instances where you have barnacles, oysters, mussels, whatever it might be that's settling on that rock structure, that's adding organic material to the sediments, potential that you might have enhanced nitrification, denitrification. Now, does the hydrology allow for those biodeposits to stay put? Is another question. But I think that there's potential.
One more question?
We see this as a long-term application for water filtration. What to do with the waste? Because you're going to be generating a lot of oyster shells. So what to do with waste? So eventually we'll be ending up with shores right now, like we see in some oceans, like algae. So then you will start seeing all the oyster shells, some of the waste.
So yeah, so if we reach so the question is, what do we do with all the oysters that overtake New York Harbor for the Billion Oyster Project? I think there's a lot of interesting things that we can do with that. We generally with oysters and oyster restoration, you don't want to remove the shell from the system because the shell is the substrate for the larvae. But we're so successful that we have excess shell. What we're doing is we can remove it from the system. We can take the nitrogen out of the tissues, use the tissues as feeds. I mean, think about where we get all of our nitrogen. Why can't we use nitrogen from nitrogen that's sequestered in oysters from New York Harbor for feed for pig lots and chickens and whatever else it might be, or fish farming, to generate feed for fish farming? There's potential for it, but I think that's a long ways down the road.
Well, thank you, Chester. A little bit about next month. We have a sort of a kind of a different kind of seminar. We have an attorney coming in that's going to be talking about the Westway project, which some of you may be familiar with. And the title of the talk is Fighting Westway, and